Okay, welcome everybody. Hope you had a good long break. It was almost like spring break, having Friday and Monday off. That's a whole lot of time. All right, not quite. So as we shift over and start thinking about how bacteria grow, I want to start by saying it's not all the same. Some can grow super quickly. Lots of them that we work with in the lab fit into that category. And, it takes them a minute to figure out their new environment. We'll talk about that in a second. But once they really start growing, they can potentially double every 20 minutes. Replication is pretty quick with a lot of the bacteria. And when we think about like infections, that becomes important. There are other bacteria, and some of them that we think about infectious, whose growth rate is super slow, like tuberculosis. That's doubling time might be a couple of weeks. So it's really a big spectrum when we think about this. Oh man. I'm going to mute all, and hopefully that doesn't get rid of me. All right. If you quit hearing me, you'll let me know, I assume. Okay. So there's all sorts of things that we think about as far as microbial growth, and there are some potential issues with some of this. This first image, it doesn't look gross or disgusting, but it's a little bit disturbing. So this is an image of some biofilm material that's on a catheter, and the bacteria is some Staph aureus. Staph aureus is a bacterium that is pretty common, something that we would expect to find on our skin. Not every square inch of our skin, but it's a normal inhabitant. And ultimately, when we think about things like a catheter situation, the potential for organisms to maybe be brought into your body is there. Aureus, a normal one on our skin, but something that we really think is being pathogenic when it's in the proper environment. Staphylococcus epidermidis is another bacterium that you'll look at in the lab. You've probably already seen a little of it, all right? It's one that we really think of as not really causing any issues, but when it comes to catheter situations, Staphylococcus epidermidis definitely can set the stage for causing problems. And with individuals with like indwelling catheters, those that go directly into the bladder through the skin, not through the urethra, it sets the stage for potentially some big issues with those individuals. The next image is a rain bucket. So we've got a big pail here with some growth, well, all over in this bucket, but a lot of it on the side pointing to some biofilm stuff, lots of algae, lots of bacteria, and in turn, given the proper conditions, they may thrive in this type of a habitat. So we've shown, and we're going to see another image of some biofilm formation on a slide with how they all start working in concert with one another to set the stage for something like that to truly occur a few slides down the road. So initially, we want to think a little bit about how bacteria go about their business of replicating. We're not going to go through and talk about the details inside with synthesizing their DNA and all of that stuff. Just this initial bit as we get started. I like this first image of some salmonella typhimurium that's in the process of replicating. So 2 cells, and we can see they're starting to separate. We're right at about this point over here for the septum, where we're starting to pinch together. It's not the same, but it's similar to cytokinesis when we think about our own cells going through replication going through mitosis or meiosis for that matter, as they pinch off into two separate cells. That's what these guys are in the process of doing. When we look at some of these things under the microscope, it can also clue us into some mechanisms that they may utilize when they replicate. Oftentimes looking at the bacillus forms or the rod-shaped bacteria, maybe not. I mean, they basically divide like this. Sometimes they stay attached and we see big long chains of bacillus, but oftentimes they're just kind of singlets all over the place, all right? But when it comes to some of the coccus-shaped bacteria, that's when we can start thinking about maybe getting some details just by looking at them, how they've stained up. The streptococci pair up kind of like these guys are doing, end to end, and make a big long chain or potentially doublets, but they're linked end to end. The staphylococci divide in irregular planes and you get big cluster shapes with them, because it's not just a set fashion, it's random truly with how the septum starts to form and you get this clustered sort of appearance. So it can clue you in a little bit about how their mechanisms of replication are occurring when we look at them under the microscope, all right? So in this instance, we can see our bacterial cell, it replicates its chromosome. There are lots of details that are happening here that we're not really, like I said, I'm not going into all of it, but when it does replicate, it's like a linked chain. That nucleic acid gets cut and separated and glued back together. There's enzymes that participate in all of those interactions. We're simplifying this stuff here a bit, but the chromosomes truly are attached, part of them, attached to part of the plasma membrane of these cells. And as the cells elongate, they separate these chromosomes to opposite ends of the cell that's going to become cells, all right? As it starts to form this septum, truly these chromosomes are pretty much moved to the opposite ends by this point. A lot like what's happening in mitosis, but there's lots of proteins pulling those chromosomes or sister chromatids to opposite ends. And this is utilizing the plasma membrane to kind of do the same sort of an idea. And then they separate. And like I said, potentially still kind of looking like they're all attached, or maybe as singlets like we show right here. So not the same again, but kind of a similar idea as these bacterial cells are replicating. In eukaryotic cells, there are other proteins that pinch and constrict to form a cleavage furrow. Not the same, but the same sort of an idea, all right? So this FTSZ ring, they're these respective proteins that basically start to pull or pinch the plasma membrane together in the center, and the septum, basically we see this forming in between cell wall material, forming in between both of these cells. This is sort of like what happens in plants, although there's lots of material, cell wall material in a plant being deposited from the Golgi apparatus. It still needs cell wall material in between the two. And in these bacterial cells, it's bringing it as it starts to fuse or pinch together. So the bacteria replicate through binary fission, and really it's like exponential. It's not like, it's exponential growth. 1 cell to 2 and 2 to 4, 4 to 8, and we just keep going, doubling and doubling until you can't double anymore, right? But they keep going, right? As this happens, it really doesn't take a whole lot of time to go from a single cell to a boatload of bacterial cells and potentially establishing an infection or colonizing to stimulate the formation of biofilm whether we're talking in your body or in the pond or whatever it might be, all right? Doesn't really take a whole lot of time for there to be a tremendous amount of bacteria that get generated. So here we can see the number of generations, 3 generations down the road. Now we've got 8 cells, all right? Starting from this first generation at 0, right? With 1 cell, all right? As it replicates, so on and so forth, all right? Lots of cells. And ultimately, these guys are like clones of the one up above, genetically identical to this initial parental cell that replicates repeatedly. They're pretty much the exact same, unless there's some subtle mutations that have occurred. Pretty much they're identical, right? Asexual replication gives us the same stuff, all right? So you don't really see this stuff in the lab, but you're doing it, all right? As you take some bacteria and put it into something else. I know you've made some transfers where you've taken some bacteria, probably from a broth, and you put it onto a petri dish and you put it onto a slant and probably into another broth, right? I think you've done all of that stuff by now. As you took that little bit of bacteria on your loop, an inoculum, if you will, as you took a little bit of that bacteria from your initial tube and made those transfers, put it into whatever else you put it in, that first little bit of bacteria goes through a little bit of time, maybe a couple of hours, where it's just checking out its surroundings, right? Just moved into town is kind of what I think. And this is the lag phase, right? We're not really seeing an increase in the number of cells. They're just getting used to their new surroundings, right? Once they've got it figured out, right? Once we've figured out all of the one-way streets in Pocatello, right? Now we're ready to do something, all right? And that's when the big burst of growth truly starts. We call this the log phase or exponential growth or exponential phase. This is when they are growing very rapidly, all right? And in this circumstance, as we're looking at it, we would think about it in whatever it is that you just transferred it into. So into that other little tube of broth that you just made your transfer into. It's a finite amount of media, finite amount of nutrients that are available for that bacterium, all right? We'll show you some other opportunities that may keep their exponential growth going indefinitely, all right? But typically, we think about them reaching a point where they level off. The stationary phase is where they've plateaued, and at this point, the birth and death rates are basically equal. We are dying just as quickly as we're replicating. We've kind of maxed out the amount of cells that this habitat truly can maintain, all right? And they've got a certain amount of time. We don't have really any numbers down here, but depends on your tube or your broth or your plate, whatever you put them in. They've got a certain amount of time that they can survive with these nutrients, and then they start depleting the nutrients and the wastes start increasing, and that's when we move to the tail end of this figure, the death phase, right? Where there are more dying than they are being created. And it almost is the same as our exponential growth. It's exponential and almost the same sort of an angle. They die pretty quickly, all right? This is a growth curve, all right? And like I said, as I started this, there's all sorts of differences when we look at bacteria. And some of them may be doubling every 20 minutes and others taking much longer than that. And understanding how they behave in their respective environment clues us into some details about that, and maybe how we might think about combating them as well, all right? There are oftentimes some labs set up to look at some of these growth curves. It's not something that we do in the introductory lab. I remember back in the day when I was taking a microbiology course, there were multiple sections, just like with you guys, starting in the morning and going all through the day, and even a night lab. I'm sure I know there's a couple of night labs for you guys, right? So we had multiple lab sections, and the very first lab set up the experiment to check these growth curves. And then every lab coming in would make measurements, trying to see how this bacteria was growing over the course of a day, right? And there were a few, taken one for the team, that showed up at about midnight to make the very last growth curve. Oftentimes, those are jobs for graduate students. They're like, you should be here, right? You can be doing these measurements, right? So we did this over the course of most of the day. Technically, you could stay up all night, right? And you could make these measurements and get 24 hours worth of measurements. We didn't go that far, but you could, right? And truly see what's happening. As we look at this, now we're looking at 11 hours. Now we've really got some time going on, and we're plotting how quickly these cells are replicating. This first little bit of time is the lag phase, where they're just figuring out what's up. And then we start replicating, and then we kick it into gear and really start growing pretty rapidly, right? And in 10 hours, we've made it to just over 1,000 cells, right? A good amount, right? If we were to continue with this and look at this the following day, we could be pushing over 1 million cells as we start doubling and doubling, or even upwards of 1 billion cells, right? We've shown this in two different figures here, because when they do start growing super quickly, all of a sudden it's just like a pretty much straight line, right? And it's very hard to extrapolate details about how much you're truly looking at, because it is so steep, if you will. But if we plot it on logarithmic paper, so every mark here is a factor of 10, every mark here, factor of 10. As we increase through here, we get more of a straight, although it's angled, a linear line versus this J-shaped curve. And it's easier to extrapolate what we're truly looking at, like maybe in between here, right? When it might be very difficult when we're looking at it just on regular graph paper. So we'll show you how we make some of these measurements with some spectrophotometers in a second. We'll get to some of those details and really think about how we might calculate some of those growth rates of bacteria. So I mentioned that what we were looking at initially was it was a closed system, right? It's just this test tube or this petri dish. It's only got a specific amount of nutrients available. And once they hit that stationary phase, once they plateau, it's not very long before we start hitting the death phase and we start dropping off, right? But we've got ways that we could keep these bacteria happy. And it really depends on some of your circumstances as far as would you want this or not, all right? When we think about the micro labs, we don't think about these situations. We don't think about needing to keep these bacteria growing exponentially indefinitely, right? But for some types of research, we may want that. Or for some other biotech stuff, we definitely might want that, right? We can genetically engineer all sorts of things, and bacteria, they're very good to manipulate and utilize to produce lots of different compounds. Human insulin was one of the very first human products that was manipulated in some bacteria And we would like those bacteria to be growing constantly, right? And we would like them to be growing exponentially so that they're pumping out ample amounts of insulin and they'd be releasing that as waste or byproduct, right? It would be going out into the media and we could collect that stuff and we could purify it and sell it, right? This chemostat gives us a situation where we could keep bacteria growing exponentially as long as it's functioning properly, all right? So the fluid in here would have all of your microbes, your microbe. Typically, it would be only one bacteria that's growing in there. You don't want to have a whole bunch of different ones or they're going to compete with each other. You're going to get problems, right? So a culture of bacteria and you're feeding it, you're pumping in nutrients and you're removing wastes, right? And if we're talking some of the biotech stuff, the wastes would be in this liquid as we are pulling it out and we could purify that out of there, right? So new food coming in and byproduct and waste going out. And the bacteria in this chamber are constantly growing because the food source is always fresh, right? So they never plateau. It's always exponential growth. And that, when they're growing super quick like that, is when they're pumping out whatever sorts of compounds because they're metabolizing very rapidly, all right? This isn't the same, but sidebar real quick, all right? If we think about in our body and the bacteria that are potentially causing infections, this is when we would like to kill them as well, all right? When they're growing super rapidly, when we see like this exponential growth and they're really kicking it into gear, they're pulling in lots of nutrients and they're trying to replicate really rapidly. And if you drill them with some antibiotics, right, as this is happening, that's when you drive a wedge right into their process of their cell cycle, right, of their binary fission. It makes them much more ready to pull in those products and for it to kill them off, right? If those bacteria are more in the stationary phase, those antibiotics are going to still work, but you're better off when they're growing really fast and pulling in nutrients as quickly as they can, because that's really when we've got a better chance of targeting, all right? So doesn't really fit with this, but it fits with thinking how they're growing, all right? So it also depends on your circumstances, but lots of times in these types of chemostats, we're thinking about aerobic situations. So we've got this little blender in there, this stir bar, if you will, that as it's moving around, it's keeping it nice and oxygenated. So those bacteria are getting everything they truly need. And if they're aerobic, they want good amounts of oxygen so they can go through their cellular respiration, right? So we're providing nutrients and oxygen and removing wastes in the process of doing it. There are also, besides chemostats, bioreactors are other names for this. It's a similar sort of an idea. And you've got opportunities to grow massive amounts of bacteria in those situations, right? And maybe that's what you need. So we look at how bacteria are growing, and we could look at them through a microscope and get some ideas about how they're growing as well, all right? Not with just a simple microscope slide, all right? This is a special type of slide. So this glass slide, and it's super thick, you pipette in a small amount of broth, bacteria in broth, into this slide. And then when you look at it under the microscope, it's got a bunch of checkerboards in there. It's all gridded off, right? And as we move into the inner bit, one little square right here is what we're looking at in this blow up over here. And you can count how many cells are in each of these, and in turn extrapolate, as we see down here, over 1 million cells per milliliter are what we're dealing with in our sample based off of this little bit of bacteria that we were able to count. And it's, as it's gridded out, it gives us the opportunity to get those details as far as how many cells we're truly looking at. And it goes through and gives you a bunch of numbers, but I'm not going to bog you down with all of those details. You count them up and you just count one of those little things, and it gives you the ability to extrapolate out total volume, all right, that you're dealing with. So there are other opportunities that we may utilize to get a gauge as far as how these cells are potentially behaving. And with this, we've got some fluorescence microscopy where we've stained up some cells. The green guys are the ones that are living, and the red ones are the ones that have died, all right? And as I look at this, my gut says we're looking at some exponential growth, right? If I looked at it and saw 50-50, half red and half green, I think we've probably plateaued, right? Or if it's much more red than green, probably in the death base, right? So you can get some hints about some of that stuff based off of that. This is pretty cool. So a culture counter is a type of electronic device that's able to measure individual cells that are moving through this device. And so there's a charge difference that basically counts as a cell moves through here. So we've got a couple of electrodes, and as the cell passes through, it breaks that contact and it makes the machine count, right? There are really some pretty fascinating details with some of these culture counters and other similar sorts of mechanisms as we are counting cells. Flow cytometers are doing a similar sort of an idea, counting individual cells. And some of these have some pretty high resolution microscopes linked right up with them that can, not only are we counting individual cells, we're taking an image of each of those cells as they're going through. And there's some high-speed photography that can make all of these images and capture it in real time as it's passing through there. That's pretty cool stuff. Coupling some microscopy up with that. So sometimes when we are thinking about trying to enumerate or figure out how much we're really working with or looking at here, we go through different types of plating to try to figure out what we're dealing with. Sometimes some dilutions, oftentimes some dilutions, to give us some details about what we're truly looking at, right? So with this, we've got a flask that's got boatloads of bacteria, but we don't know how much, right? You could potentially use that little slide and count some cells, or we could do some plating and look for some colonies, some growth on these plates that might give us some details as well, all right? So we've got a fixed volume of cells mixed with some broth that's going to do our diluting, right? And really the idea is to try to figure out how much we're really looking at, right? So we take a milliliter, maybe we deal with different volumes, but in this case, 1 ml added to 9 mils of broth. There's nothing, although it looks turbid here, all right? As we do this first dilution, 1 mil into 9 mil, there's 10 mils total. This is a 1 to 10 dilution. And we take some of that, plating it out. And this plate is just covered with bacteria. You can't see any space really in between one another. It's just kind of a lawn of growth, too numerous to count, right? we take a mil of that into another 9 mils. Now it's a 1:100 dilution. We take an aliquot of that and plate it, and it's still, although I can see some gaps in there, still super thick and very difficult to really say what's an individual colony in the first place. All right. So another too numerous, another 1 to 10 dilution. So this third tube is at 1 to 1,000, pretty dilute. All right. And as it's plated out, and it's 1/10 of a mil, 100 microliters that we put in there, right? As we plated this out, now you technically can count it, all right? 389 colonies is a boatload of colonies and difficult to count, all right? But you could, all right? Oftentimes, there are like some light boxes where your plate goes on there and you can make some counting. Sometimes there are some pens that have a counter attached with it. And as you touch the plate, it's also counting and keeping track. And then you know, right? If you're just visually looking at it, counting 389, are you sure you counted them right? Maybe, right? If you hand it to your lab partner, they may end up with 350, right? And then you hand it to somebody else and they get 400, right? The little pen touching each colony makes it so you're pretty sure you counted it accurately, right? This still pretty difficult, all right? But you could technically count them, all right? This next dilution, now we're getting better, all right? So at a 1 to 10,000 dilution, all right, we plate this out. Now we got 50 colonies. That's pretty easy to count, all right? And then our very last one is we've diluted at 1 to 100,000, 2 colonies, all right? I can count that for sure, right? But I feel a little more comfortable dealing with this, all right? Where I've got more than 2, all right? Either way, all right? We can look at this with our respective colonies, knowing I took 100 microliters out of that, and it's a 1 to 100,000 dilution. And now we're talking millions of cells, all right? As we looked at before with that 1,250,000 cells, similar idea as we're looking at this with the 1/10 of a mil as our amount we've added, right? This is, it's not the same, but it's a similar sort of an idea. Instead of taking just that little bit of bacteria and putting it onto the plate like we were looking at before, here we're taking a small amount of bacteria and we're adding it to some agar, some media that is warm, but not cooking hot. All right. When the agar is between 45 and 50, it stays Celsius, it stays liquid. All right. And once it's poured into this Petri dish, it really starts to cool pretty quickly. And the bacteria that's added to this, it's not so hot that it kills them. All right. And the temperature drops pretty quickly and then they're good to go. All right. So the bacteria gets mixed with this, all right, and then you pour it into the Petri dish, all right? There's no agar in there yet. It's all in this tube, all right? So you pour it in there, you swirl the plate up a little bit to spread all of it out, and then you allow it to incubate. You would have a handful of pour plates, handful of dilutions, if you will, that are similar to what we're looking at here, all right? And as we allow it to grow, now we can start seeing some individual colonies, all right? And counting it up based off of the same sort of an idea. How did we dilute these to begin with? This is another idea, and this really, the Petri dish in number 2 should have some agar in there already, all right? And this looks like it's got nothing, all right? In a spread plating technique, the Petri dish already's got some in there. We take a small amount of bacteria in broth and we use this spreader. We call it a hockey stick or a glass spreading rod. And we spread it around on the Petri, on the agar surface. And you would do the same. There'd be multiple dilutions, each one getting their own plate, and you should be able to see some colonies growing on there. The idea is to dilute stuff out enough that you can count it, all right, in all of these circumstances, all right? And this is a pretty good method to get the bacteria spread about, all right? We will think about, potentially see some stuff in the lab where we look at antibiotics to treat specific bacteria. And you take a small amount of bacteria in broth and you would spread it all over this big petri dish, and then you'd apply antibiotics to it and see, did they kill the bacteria? Did they inhibit the bacteria growth or not, right? And in order to spread it all out on there, it's the same spread plating method to get them all in there, but we're not diluting it. We're just spreading it all over, all right? So here it's another idea with some dilutions, and we're looking at, in this case, most probable number method. We've got a handful of ferments, so lactose broths. Have you done any fermentation stuff? Have you seen any of these tubes? Okay, you will see some of them, but you're not going to do the same idea. So when we look at these tubes, there's a sugar, and in this case, it's lactose. You're going to have a handful of different types of sugars and see, can these bacteria use it or not, all right? So in this case, lactose is what we're looking at. And if the bacteria can utilize it, fermenting the lactose or the other sugar, whatever it is that might be in there, the media goes from a red color to a yellow color as the bacteria alters the pH, all right? It's kind of hard to see, but up here, there's a little bit in there. Down below, you can see it because we've turned it yellow. There are some other tubes that are flipped upside down in these ferments that if the bacteria is growing in there and it's producing gas in the process of fermenting, it gets trapped in these little tubes. They're called Durham tubes. It gets trapped in there, and then you can see that it fermented, changed it to a yellow color, and it also produced gas. Some bacteria only ferment and they don't produce gas, and the little tube inside of there won't have an air bubble in it. It'll be still full of liquid, all right? But if there is a bubble in it, you know it's produced some gas, all right? So here in this case, right, they have basically done some dilutions as far as how much we're putting in here, all right? And seeing what happens over the course of time, all right? So looking to see how many are changing. So 5, 2, and 0 positive tubes over here, and this probable number, not something that we really think about with the stuff we deal with, but for maybe some environmental, as we're looking here with some pond water, it might give us some better opportunities to extrapolate what we're really looking at. So the growth curve that we kind of started with in here, we could take our bacteria, we could inoculate it into some broth, right? And then we could allow it to grow over the course of time, and we could make some measurements and see what's truly happening, all right? So a spectrophotometer is basically shooting some light at your sample, and it's measuring, well, it can measure multiple things, absorbance or transmittance. So it just depends on the specifics. Usually we are thinking absorbance. And so before you do the inoculating, you take your broth that doesn't have any bacteria in there, and you read its absorbance in the spectrophotometer, right? And then you inoculate it, and we start making measurements over the course of time. You need to have one that doesn't have anything in it so you know what your 0 point is, right? And then as the bacteria starts to grow, that broth goes from see-through, I'd say colorless, but it's yellow, goes from see-through to turbid, right? And as the light is being shined in there, a good amount of it gets reflected away, and a little bit gets transmitted through. And so we are seeing a decrease in the amount of light that's moved. And we're trying to show that with this big thick bar and a little thin bar down below, right? There's less of it that's able to move through. And we make these measurements, right? And with that, if you're doing it, making measurements every 15 minutes or every 30 minutes, if we think about bacteria replicating every 20 minutes, potentially, maybe you would like to make measurements about every 20 minutes, right? Over the course of 12 hours, potentially. That's a lot of measurements, right? Think of all the data points. All right. And that in turn gives you the opportunity to maybe see it stationary and maybe even get the hint of some death phase as it moves through there. That's the idea, right? And really kind of depends on the circumstances as far as what we might utilize. This looks like just broth in a test tube. There are others where it's a flask that's got a test tube attached to it. They're very different looking flasks. They're called nephlometer flasks. I think they probably have another name as well. But it's a flask with a test tube poking off the side of it. They go into a big incubator and shake it up, but it basically makes it so that your tube that you're going to put in the spectrophotometer is attached to the flask. You don't have to do any pipetting. You don't have to do anything with it. It just goes in there. You measure it before any bacteria, and then you just pull it out every 15 or 20 minutes, make the measurements, and put it back in there, right? That makes sure that everything stays clean. You don't potentially contaminate it with other stuff over the course of a day's worth of readings, right? Pretty interesting. And probably pretty thankful that you don't have to do that type of stuff, I would think, all right? Takes a long time to not really see much of anything happening, right? You hit exponential and then you're seeing it, but it's really not that impressive, all right? Okay, so we've got a couple of other images as we look at this with some other ways for some replication. So some filamentous thread-like sort of cyanobacteria. These guys replicate by fragmentation, breaking off little pieces of that. And then we've got another bacteria over here, Gemata. These guys replicate through a process called budding. Yeast cells do some similar stuff like this, and that's what we're looking at as we see those guys. So this image, we saw this guy before. I don't remember what chapter it was, but I know we've seen it, right? So some biofilm formation as we see this substrate, whatever it might be, some surface, maybe that catheter, right? So we get some planktonic cells, free floating, free living, that can attach to this substrate, all right? And at this point, they've got some potentials to maybe not stay attached. They're reversible, all right? But this is a pretty quick thing, all right? After a few seconds, these colonizers basically kind of get glued down, all right? Their sugary coating, this glycocalyx layer, helps to ensure that they stay put, if you will, all right? And at this point, after a few seconds or minutes of being on this surface, their secretions, this capsule layer that they have produced, stick some to the surface, all right? And then they start growing and dividing, and this takes some time. Now we're moving from minutes to hours or days, and we progress, right? As they keep doing this, there's more of the secretion to the outside, making it protected a bit on the inside, making it difficult for maybe some drugs to come in and target biofilms that have formed in your body. And then we've got some potentials after prolonged time, all right, where maybe some of these start to disperse. We've got some planktonic organisms moving away from this biofilm to potentially colonize elsewhere and start a new biofilm, all right? There are lots of details as we look at this participants in here, where we think about maybe more of a community sort of situation, not just one type of bacteria, although some would be single type of colonist that forms this biofilm. Oftentimes there's multiple participants and they kind of feed off of one another, all right? Where some of the byproduct from one becomes the input of the other and they work together, right? And form this community. And it's a win-win for them, right? And maybe not for us, right? Kind of depends on the circumstances. So I don't want to get bogged down with this stuff, but there are lots of things in this community, right? Lots of things where these organisms are participating and potentially communicating with one another, all right? Sensing things in their environment. They can sense nutrients. They also can sense one another with different molecules and potentially communicate based off of some of that, all right? And that's this next bit, maybe. This next bit. Buttons and mice, what do I do? All right, so this short peptide is something that bacteria can use to sense others, all right? So some gram positives and some gram negatives. Gram positives using this, and then this N-acetylated homoserine lactone with the gram negatives. You don't need to get bogged down with the biochemistry behind these molecules. They're just things that the bacteria can smell, if you will, all right? They know. And we call this some quorum sensing, all right? As the bacteria are in there and growing and replicating or participating in this biofilm, they can start producing, secreting these molecules that the others can start recognizing. And potentially, when there's enough of them at this point over here, or maybe as we're right at this, you know, 0.4, as we start getting enough, that's when these molecules start getting secreted and they start sensing it. And they're like, okay, there's enough of us. We're good to go. Maybe we should move, or maybe we should stimulate disease production if we're thinking inside of our body. And some of these colonists that have taken hold and then replicated to the point that their numbers are now sufficient to actually say, we're good to go. And we could really establish disease at this point, all right? The first bit is just the initial colonization. And then their numbers are plentiful enough to truly say, let's do something. And it's these molecules that they sense that basically says our numbers are high enough, all right? Which I think is pretty impressive, all right? And that's a good stopping point. We'll pick up with the rest of this chapter Friday. Farewell. Good day to all.